Hi, I'm Amanda McGill-Johnson, the Executive Director of Nebraska Coalition for Life-Saving Cures. Our mission is to promote, advocate, and support medical research and health science research for the betterment of our economy and our personal well-being. We have just started a new program, Talking Research. This is the fourth time we've done it. And the goal of this program is to bring in just awesome scientists from around Nebraska to talk about the incredible work they're doing and to just raise awareness for this work. So we are super fortunate to have Dr. Angie Pannier here with us from the University of Nebraska-Lincoln. She's the Maxcy Professor of Biological Systems Engineering there at UNL. Thank you so much for joining us, Dr. Pannier.
Sure, thank you for having me. I'm excited to chat.
Awesome. Well, and your work, I'm trying to wrap my head around the work that you do. You focus on gene delivery. The everyday person may not know exactly what gene delivery is and means and what it could potentially mean for their health. So can you talk to me a little bit about what you do in terms that maybe you would share with your neighbor?
Absolutely. So many of us are familiar that we have DNA, our genetic code that you're going to find in the nucleus of your cells. And that genetic code famously controls things like your hair color. Well, sometimes our hair color, maybe not always, our natural hair color, our eye color, our height. But really, those genes are pieces of information that help our cells function every day, every second of every day, and do things like heart cells to beat, and intestinal cells to allow nutrients to transport. And so those cells are become who they are and control their functions because of information that is contained in genes. And so what happens is in some diseases, we know that that book of information, that gene, might have some sort of mutation in it is the word that we would use, or some sort of problem with that book that then produces a protein that it shouldn't be producing that can cause a disease. And so in gene delivery, our idea is to deliver a new copy of that gene or a new copy of that book that helps to correct the mutation and enable a therapy for the disease. Or we may have a cell that just won't read that book, doesn't want to look at that gene. And so we're going to deliver that gene again and say, no, look at this copy, because you need to be reading that book and using the instructions in that book, which typically go on to instruct the cell to make a protein to help with the therapy. And so it's generally in the area of gene therapy that you want to deliver genes. And I think that it's a crazy way to think about medicine, because while we have many ways to treat diseases, gene therapies has the potential to cure diseases. Because
it really gets to the heart of
it really does. And so famously, things like cystic fibrosis, hemophilia, and there's many other diseases that we know the genetic mutation causing the disease. We know the book that has the error. And so if we can deliver the correct book, then we can go ahead and cure the disease. And there are now, when I started in this field, we dreamed of the day that we would have FDA approved therapies. They are now several FDA approved therapies that make use of gene therapy to cure a patient. And then I'll say one more thing, and that is that our cells look at the genes, they read the books, but they use an intermediate molecule to actually make the protein, and that intermediate molecule is messenger RNA. It looks a lot like a gene. It has the same sequence as the gene. And so sometimes we're also interested in delivering that instead of the whole gene. We are interested in delivering the messenger RNA. And the reason I bring that up is because things like the Pfizer and Moderna COVID-19 vaccines deliver messenger RNA. So gene therapy is having not only a renaissance, but it is great to be in this field suddenly, where we've worked so hard to have a technology now that we have administered to already millions of people and are keeping them out of the hospital and safe from COVID-19.
Yeah, it's technology that's become a little more household friendly.
Okay, yeah.
It's like, I've heard that somewhere. Oh yeah, with those vaccines. Now, are these genes sometimes, are they taken from an individual's person's DNA, or are they?
They don't have to be. So, DNA and genes really is a string of chemicals that we know how to synthesize and make in the lab. And sometimes we have to use an intermediate organism to help us make lots of it. But typically we don't have to go in and pull it from the patient. We already know the sequence we want to deliver. One other thing I'll say is that there are some cases where we actually want to take our gene, that book, and click it into our genome so that it's permanently there. And that can be a little scary for folks, and I agree. But there's a lot of ways that we actually don't have to have it clicked into our genome. We can have it sit as a separate book by itself, and then that way you don't have any concerns about sticking it into the genome. But
yeah, that gets people, just a lack of understanding about how that basic
Absolutely, yeah.
science works and what people feel comfortable with. If it can work, then just attach it.
Absolutely.
So I understand from trying to do a little homework on your research that there are viral and non-viral ways to deliver genes into cells and tissues that need them. What's the difference? And talk to me about the effectiveness.
Absolutely. So we have to get this book, which is really, this piece of DNA. It's a long sequence. We have to somehow get it into a cell, and not only into the cell, but into the nucleus, so that that gene exists in the nucleus of the target cell. And so if we use viral vectors, it's really taking a virus that we know can infect cells, but we have really smart ways to engineer out the parts of the virus that make it a virus. So it can't be infective, it won't make you sick, and it can't replicate. Viruses can replicate. That's why we have a pandemic, because SARS-CoV-2 is replicating all over the world. So we take out all those parts, but what we leave in that virus is the parts that help it shuttle genes into cells. That is what viruses do. And so that is, and it's very effective in some cases. But there's issues sometimes with scale up growing lots of viruses and engineering it out. Also, it is still a virus. And so if you deliver it to a person, they'll actually form antibodies against the virus, even if you're just trying to deliver a gene to cure cystic fibrosis, but they'll develop antibodies to the actual viral particle that you're using. So you can't always repeatedly dose individuals there. So there's some shortcomings with viruses, but they are really, really effective. And famously, the AstraZeneca COVID-19 vaccine and the Johnson & Johnson COVID-19 vaccine both make use of a virus to deliver the gene into our patients. And it's using a cold virus that has been engineered to not cause cold at all. It's impossible. It's not even there. It's just using the shuttle part of that. I work mainly in non-viral gene delivery, which is where we use synthetic molecules and sometimes natural molecules to form particles that mimic a virus, but they are in no way a virus. So these oftentimes in some instances can be safer, have less of an immune response, so we can repeatedly dose it. And there's a lot of cool ways we can I'm an engineer, so I love to make little beautiful particles that have genes inside of them, and we get to do that. But nature is still better at delivering genes, so they're way less effective than our viral I guess challengers are, because I am in the non-viral world. But so there's advantages to each, and you can see that playing out, because one of the most famous ways to use non-viral gene delivery is to use a fat bubble to encapsulate a gene and deliver it into cells. And the Pfizer and Moderna COVID-19 vaccines use a non-viral gene delivery, a fat bubble, to protect the messenger RNA and to get it inside of your cells. And so you can see that both of those vaccines are effective. They really do work a little bit in different ways. But I think there's room in this space for us to keep engineering both types of delivery systems, both viral and non-viral.
So you've gotten several national awards for your work over the last 5 years or so. And your pioneering research really focuses on preparing the cells for that gene. Can you talk to me about that?
Yeah, so I am an engineer, and I do like designing little nanoparticles, little fat bubbles and other things like that to make them more potent so that they can compete with a virus. But my lab has really taken a different approach since I started in this work, which is, okay, we can design a nice little nanoparticle, and I'm going to keep doing that. But we have to remember that there is an active role by the cell. The cell has to enable or allow that fat particle to go inside of it, and then to get the gene inside the nucleus. And so a big part of my lab, we actually think about, can we do something to make that cell happier? What we say is to make that cell more receptive, more agreeable to taking up the genes. And so we do things like treating it with pharmacological agents to help it sort of take on the stress of having genes delivered to it. It can be kind of stressful for a cell to have this DNA put into it, and that stress can sometimes limit your efficiency of how many genes you can get in. So we've pioneered this approach that, what if we prep our site, prep our cells, and then deliver our beautiful little nanoparticles with genes in it? And we've shown that to be highly effective.
That's terrific. And in the title of today's discussions, we've talked a little bit about vaccines. We also talk about stem cell therapy. So talk a little bit more about, how this technology is used. You've mentioned the vaccine piece quite a bit, but also
Right.
stem cell therapy. And for people who might not be very familiar with the concept of stem cell therapy.
Absolutely. Well, let me touch just a little bit more on vaccines because it's so relevant and so much,
Oh,
and we work on it as well. And so, what we had envisioned in my lab long before it was cool and all these COVID-19 vaccines have come out using this technology is that old scale technology for vaccines, you have to have that pathogen. You have to have a sample of your influenza virus or your pathogen, and then we grow it up. And it takes a long time for us to prepare for those vaccines. And we have to have a piece of that virus. And so there could be safety concerns growing it. And that's case in point of we always have to predict our future influenza strains 6 months in advance, mainly because it just takes us that long to grow them up. And so many of us in the field thought, what if we get rid of the pathogen altogether, go in and take a gene of the pathogen? These books of life are in every living organism. We'll take one of their genes and deliver it to our cells, and then that they can act on that gene as though they were infected by the virus or pathogen without a virus or pathogen ever being with them, making it safer, but also scalable, and especially in the event of a pandemic. And so that a big part of my lab does work on DNA vaccines, which is what we're seeing played out. And I do think that with the COVID-19 vaccines that are approved for emergency use, we're going to see a change in vaccine technology for the better for humankind. It's going to be very, very cool to watch to see the evolution of how we can vaccinate our individuals. In terms of stem cells, so we also, in addition to delivering genes for vaccination in my lab, we're interested in delivering genes to stem cells to coax them to do things that might be important therapeutically. So stem cells are cells that are found, there's many different types of stem cells. There's some that have ethical and moral challenges. These stem cells that I work on in my lab are adult-derived stem cells. So they're found in your bone marrow, in your fat tissue, and we actually collect them in my lab from urine because they're actually being shed off the inside of the bladder.
I had no idea.
I know, we do collect it. And that's a really easy way to collect stem cells. Even fat, you get liposuction and then we can collect some stem cells from you. And the idea is that these stem cells sit in reserve in your body, and their job is to then mobilize if you need to be repaired in some way. What they can do is while they're a stem cell, they kind of don't have an identity, but they can become whatever they want to become. So these stem cells can become fat or bone or cartilage and other things that they can become. But what we've also found is that these stem cells, and when I'm talking about these stem cells, I should say they're mesenchymal stem cells, they themselves just seem to be mediators of wound healing and do some what almost people seems like magical properties. And so there's a lot of clinical trials, even in COVID, where you take someone's mesenchymal stem cells from their bone marrow, from their fat, you purify them out, and then you give them back to your patient, and they actually home to sites of injury and inflammation, and they can do all these kind of cool things natively. But what we want to do is deliver genes to these cells to coax them to do other things as well. So if we already know that these stem cells will migrate to a tumor site, we already know that, why not give them a gene that allows them to secrete a factor to kill those tumor cells? No. So then they get to that tumor, and then they start to secrete that factor that we've genetically engineered them to do to help treat that therapy. The other famous thing about these cells is they have been shown to cross the blood-brain barrier. So the idea that we could treat something like Alzheimer's with these cells, they're going to hone to the site of inflammation in the brain, but then we've engineered them to secrete some enzymes to degrade plaques or to promote neurogenesis. And so that's our idea with these stem cells, is to capitalize on their innate properties and then endow them with some better superhuman properties. Maybe we shouldn't say that, it scares people. But it really would give them, the ability to be super targeters of therapies.
With as quickly, like I think people are all just in awe about how quickly the vaccines were developed. And people, and that's been one of the struggles too, right? Getting people to feel comfortable with it, not because the general public didn't know this technology has been in development and it was ready to go to meet this need. Right. Now that we have this technology, how much quicker do you think science will move to being able to treat people and find new solutions, maybe a little quicker than we have in the past?
I think we are going to see some therapies come out maybe quicker than we had expected. And so, I do like to say the messenger RNA technology and the fat bubbles we're using to deliver them, this is not in our field. We've been studying these fat bubbles for 30 years, and we've been studying messenger RNA as a delivery or as a therapeutic. In fact, it was in clinical trials for an influenza vaccine. I mean, we were ready to go, and that's the value of funding research and basic research, because when we needed to go, we were able to go. The other reasons it went really fast is that we put a ton of more money into it and got it cured, basically, right? Solved. And so I don't know if we're going to be able to invest $15 billion into everything, but if we did, we would be able to have medicines come out very quickly. And that shows you that. But with that being said, one of the things you have to do is to prove safety with a technology. And we now have, gene therapy, whether it's through a adenoviral vector with the Johnson & Johnson vaccine, or whether it's with a fat bubble and messenger RNA with Pfizer and Moderna in millions of people. So you can already go to the FDA and say, well, we know it's safe. Now we're going to switch out the book. So rather than messenger RNA be targeted for COVID-19, now it's going to target for cancer, or now it's going to target for, some other thing. So I do think we're going to see other medicines come out. I will say this big asterisk though. You don't have to get very good transfection is the word we use when you actually successfully get your gene into a target cell. You don't have to have that good a transfection to have a good vaccine. You have to have pretty good transfection to completely kill a tumor. You have to have a good transfection to completely cure a genetic disease. And so, but we still now have a baseline where we can get it to work good enough for a vaccine. So now let's get it to work good enough, for a therapy, for a disease that we want to treat.
This is more of a follow-up question on the vaccines and the technology. One of those concerns that I hear floating around out there is that, we don't know what side effects may really happen with this vaccine down the road. Can you speak to that at all? And if this technology really allows for that, or your just general thoughts?
So, there's a couple ways to address that. First of all, I want to say that I think there's some misunderstanding that we have long-term data on every medicine that you take. Every day there's a new commercial with a new drug that we don't have long-term effects of that. We have what we thought was a long enough effect in a clinical trial for the FDA to approve authorization, but we don't have 30 years of data. Those drugs were just brought out. And so there is a misconception that we have some long-term data of everything else except for this one vaccine. And so that's one thing. Secondly, we do know a lot about how vaccines work. And so while this is a new and different technology, we still understand what we were trying to do and what we should measure. And then a couple more things I'll say is most of the side effects from a vaccine are going to be very short-lived. We would have seen that over and over again in clinical trials and now in mass, vaccinations, and we're not seeing that. And this messenger RNA that's in the Pfizer and Moderna vaccine is this intermediate molecule between a book of genes and a protein. It's making a protein. It is so fragile. That's why it has to be in a fat bubble. It's so fragile. So it's not hanging around in your body. It's just there as a recipe for your cell to look at it and say, oh, okay, that's a message encoding for the spike protein. I'm going to make the spike protein. That messenger RNA gets degraded, like all other messenger RNAs in your cell are constantly being degraded. I'm going to make that spike protein and show it off to my organism, to my host, and then we're going to make antibodies against the spike protein. And then at the end, you're left with antibodies against the spike protein. And so the last thing I'll say is if there's any long-term effects of that, that's going to be the same long-term effects you would have seen getting COVID because you have antibodies against the spike protein. Oh, except you're not going to have all the side effects of having COVID because that's really scary. We do not know the long-term effects of a COVID-19 infection, and we're worried about it because just the shorter term effects of 6 to 12 month long haulers, stuff like that, that's way scarier than a fat bubble with a really fragile piece of messenger RNA that was just in your body long enough to instruct your body to make the spike protein, which also didn't stay around very long, to instruct your body to make the antibodies. But you have to connect a lot there.
That's very terrific and helpful because I know some of those long-termers and it's very unpredictable.
Very unpredictable.
COVID has been. How has the pandemic impacted your work, generally speaking? I know some labs have had to be closed and things like that. How has it impacted you?
So we shut down as well, and that certainly impacts a research lab. You can't do anything if you're not in the lab.
I think behind, like in their career path too, I would imagine.
That to me would be the number one effect. No student that works on their PhD for 5 years in a lab expects to not be able to be access to their lab for a significant amount of time. So that impacted our research in a negative way. I'd say in a positive way, and just science does better when we're around each other and talking and coming up with ideas. And so that also impacted our lab. I'll say in the positive, we were able to move into a new building during the pandemic. We were able to move into a new lab space. And usually when you move, you lose time in lab, but when your lab's already shut down, they let us move when we were shut down. That was a good use of our time. Our instruments were already off. The other thing is that we do have, we're actually have a funded grant right now on developing a COVID-19 vaccine. So it let me, sort of add something to my research portfolio that I wasn't planning on working on. And we are actually working on a different type of fat bubble for oral vaccination. So
that would be terrific. I think of my little kids and how they're not looking forward to when they're going to get it, but once it's available for toddlers, they're not looking forward to it.
Absolutely, yeah. So I think that, there's been positive and negative things, but I think the other thing that I'll say for researchers, especially any researcher or any human, right, that was home with children during the pandemic, research moves about 2 years at a time. And so a lot of times what we're publishing now, we really did 2 years ago, but it takes 2 years for it to come out, or we worked for a grant and then we had the grant. So I think you're going to see a lot of decreased productivity for some researchers like myself 2 years from now, because that's when the delay happened.
I know there's been a lot of concern about women in STEM and in science and the impact as caregivers during the pandemic, when there's already a need for more women and what that will do. What do you hope just is your ultimate success or outcome of your work?
I always will say that I'm an educational research institution. So my number one outcome and impact are the students that are trained in my lab and that go on and do great things. We actually have two students, two former PhD students that are both working at a vaccine company right now, and they're busier than ever. I have another former PhD student who is one of the biological directors at the FDA overseeing all the plants for COVID-19. So they are really, truly making an impact. And so that is always going to be my biggest impact, and why I do this work is because we do really cool research. We hope that some of the systems we're developing for gene delivery can make it to the clinic or make it to some sort of a diagnostic assay. We'd love to see stem cell technologies take off, but I'm really proud of my students and what they have and will continue to keep doing.
So clearly that mentorship is really important to you. Tell me a little bit about your origin story and what got you interested in biomedical engineering. I imagine there aren't a lot of women in that field.
Well, there are a lot of women now, so it's one of the best for engineering. Yeah, there's actually probably almost 50% at the undergraduate level in biomedical engineering.
Terrific.
Yeah, and so and I was the first female faculty member hired in our department, and first to earn tenure and be fully promoted. But I am now one of, I think, 8 or 9. So it's been fun to watch that happen in my career. So I originally am from Fremont, Nebraska, and grew up really loving math, science, and physics. I'm a first generation college student. I didn't know what I wanted to do. I was smart, so everyone told me to be a doctor. And I said, I don't want to be a doctor. I don't like touching people, and I definitely don't want to touch sick people. But I wouldn't mind helping sick people. I just don't want to have to touch them. And so with that being said, I started to realize that engineering is this general field that you can use math, science, and physics to solve problems. And it's super hard and challenging and fun. And there aren't a lot of women. A lot of this was motivating me to go. But then I found out that there was biomedical engineering where I could work on medical problems and really understand medicine, but not be a clinician and impact the field in a different way. And so I was pretty hooked on it once I heard about all of this. I will say a turning point for my life and my career. I thought I would get a 4-year engineering degree and go get a job. But after my sophomore year of college, I got a summer research position at the University of Nebraska-Medical Center in the Department of what was pharmacology. It's not called that anymore, in Pointer Hall. And I had a research experience there, and I had no idea about academic research or medical research. And every day I was learning, and I was having so much fun. And I remember one day they told me to go to the library and read, and I was like, oh, because I clearly had no idea what I was doing. And I said, okay, is there like a timesheet that I can sign out of? Because if I'm just reading at the library, I shouldn't be getting paid for that. And they're like, you get paid to learn. And I was like, does everyone know about this? Like, I mean, this job is for me. And so I became fascinated, and I wanted to know, how do I become a principal investigator, which is what we call ourselves when we're running our labs. How do I become a PI, and what do I need to do? And I was just hooked on that life because I get paid to learn, and I get paid to help others learn. And we also get to do cool experiments and cool creative things. And so that was sort of set me on my path at that point.
That's amazing.
Yeah.
And I love your energy around it too.
Yeah.
Yeah.
Well, I have now trained over 45 undergraduates in my lab because I understand how important that undergraduate research experience is. And as many people love it, we also have students that come in that are like, oh, I don't like this. And I'm always like, that's fantastic, because now you don't like it.
Absolutely. Like, I'm a big believer in getting people internship opportunities or those chances to visit so they can get a sense of if it's for them or not.
Absolutely.
I originally went to UNL to go into physics and ended up doing journalism. I'm like, I want to talk about science. I may not be the best scientist. I can appreciate that. What are your pieces of advice you have for young people who may be considering a career like yours?
Well, I would say the first thing is, if you're really young and you're still in high school or middle school, take all the math and science classes that you can take, and really love it. And so sometimes I didn't talk about another part of my lab today, which is we also do tissue engineering. So we try to encourage those stem cells to grow into an organ that could be used for transplantation. It's a field that's had a lot of challenges and delays. I have so many students that say, oh, I want to do that so badly. I want to see a beating heart growing on the lab bench. I go, okay, do you like math? And they're like, no. I go, yeah, you do have to be an engineer. You have to really love math, science, and physics. So take those classes, find ways to interact. We have a lot of high school programs actually in the state of Nebraska where high schoolers can engage with the lab. I have a high school student starting in my lab next Monday. I think she's my 8th high school student that I've had in the lab. And so, find ways to do that. You don't have to, but if you're interested. And then, find a good undergraduate institution that has engineering. You can do biomedical engineering as a biomedical engineer. You also can do it through other types of engineering disciplines, and get into a lab. And, I have freshmen that have been with me their whole time. As a freshman, they start out, they don't know enough biology to do a lot of things, but they start by washing dishes and doing some of the general maintenance. And by the end, they have authorship on a paper. And so I think, and don't be afraid to talk to professors when you go to college. Like, we're just people and you're never going to get a position in our lab if you don't talk to us. And so, yeah, I'd say those are and then the other thing I'll say is time management's a big thing. So when you go from high school to college, you need to manage your time. And the other thing I'll say is that grades do matter. So work hard to get your grades up, because I have many students that fall in love with biomedical engineering as an undergraduate, and they want to go to graduate school, but they kind of had, some rough semesters because maybe they didn't make the best choices, or, maybe they didn't seek out help early enough. And it's hard. I mean, if you want to go to graduate school, grades do matter. So it's kind of tough love there, but it is good advice.
I mean, that's a competitive field, as many careers are. So it's good advice no matter what. Well, we're coming up on 30 minutes. So is there anything else you'd like to share with our viewers?
I don't think so. I just think, I guess I say I don't think so, and then I keep talking. I'm such a professor. Oh my gosh. I would just say that I think that we're so fortunate as a state of Nebraska to have the research institutions that we have here. COVID-19 again showed us we have UNMC, a world leader in infectious disease. And so what a great state to be in, with a lot of research going on, a lot more than people even realize. And also, if you want to know more what's going on, reach out. We'd love to have people visit. We can have people on campus again and show off labs and tours. And, I think that it can demystify science if you can see it a little bit more up hand and up close.
Absolutely. Well, thank you so much, Dr. Pannier, for joining us. And thank you to our Facebook viewers and viewers who are tuning in later on YouTube. Take care.
Bye.